Hello, everyone, and welcome back to Small Talk. I'm still John Tigges. We're still with Alfonso Blanco and Jenna Rogers. So this is number 2 of the 5-part series. A couple of things I want to remind you is if you have any questions, please feel free to post them. We're going to get to those in our last session. Um, you know, we're going to do a kind of more of a live portion to that so that we can directly answer your questions and help as much as possible. So today we're going to talk a little bit about isolation moving into detection, so we can talk about the thing we know, cytometry. And so I guess as a recap, my friends, is that last time we talked just about starting the experiment and all the heavy lifting that needed to be done. So there was a lot of fun to that as far as the overwhelming nature of becoming a cytometrist new to this world. So I guess we can go through and kind of just give some points of the major bullet points of what the first session was about, in case you missed it. So first was the discussion of changing your thought process. You can't walk around with blinders on. You have to be open to a lot of different things and ways of looking at it. Literature, dealing with literature searches and figuring out, what mistakes have already been made in the world so that you can start from a happy place. And then, Alfonso touched on biocontamination. If you were talking sorting, I believe, Alfonso, there was also lots of contaminants as far as, watch out for bubbles, which we're going to mention a lot. Watch out for any imperfections you can have in your solutions, your sheath, you name it. If you're doing it in the experiment, then make sure that it's as clean as possible. Any mistakes that you think you've made, unfortunately, keep making them. Because at least then you can compare the data. So, I know it sounds weird, but if for some odd reason, you decide that you're going to, chill your blood, or you mixed it too much, and keep doing it or start over. Depends how deep you got, right? If it's at the beginning, start over. It's probably easier. So any other comments, my friends?
Just to remind, what kind of samples do you have, Jenna?
We were talking about in this situation that we were looking at blood samples, and then that we were going to isolate them and use the serum. No, plasma. We're going to use the plasma.
Plasma.
Yeah, because there's less issues.
Yes. Serum contains a lot of platelets, and if we're looking at red blood cells, we don't want a lot of platelets. We actually, I was having a discussion about that yesterday with Yanitza, because we were looking at a paper that used serum, and there was a lot of things they were that we know not to be true that we saw and figured, okay, they didn't take into account the platelet contamination. Oh, that was the other thing that Alfonso pointed out greatly, is that you can use dump channels. So by labeling things that you don't want, you're able to separate out and get down to a better story, more of an individualized assays, so that you can have the EVs of interest and get rid of everything else. I think we compared this to doing stem cells, and how you have the SLKs, so you get rid of the lineage, positive population, so you know what you're looking at. But we also said that in that particular instance, so that no one gets the wrong idea, is those people publish their paper, which is slightly ahead of us. So we can only knock it so far. Some of that becomes a jealousy factor, I guess, right? Because we can tear it apart afterwards, but the only reason we get to see it is because they published. So so there you go. We learned a lesson. It doesn't matter as long as you can convince somebody else that what you say is true. So
Correct.
you just got to be a better salesman than a scientist, I guess.
That's how that would go.
Just kidding, people. Just kidding. So let's talk about isolation methods. Let's dive. We're going to take the deep dive into isolation methods. Now, we know there's plenty of position papers on this, lots of reviews. So every time you hear it, I was listening to, last week, John Nolan, different things. And as he said, the gold standard is still centrifugation in this form of ultra centrifugation. Other things are creeping in, but a lot of labs do that. As we started this discussion previously, we've had this back and forth. I guess the biggest problem is not everybody has access to one. I know in our situation, our group has one and many others share. So it's actually, feasible that way because they're not cheap, and we're all from academic institutions, which, basically spells not a lot of money. Share as much as possible, right? That's why we're cores, is because we're shared resource facilities. It's easier that way for everybody because no one wants to dump all their money into, having their own separate instrumentation. I agree because it's better to pay people than it is just to buy a bunch of instruments. Because then who's going to use them? You need people to use them. So,
And maintain them.
Yeah. So any take on that? I know, Alfonso, when we were kind of talking previously, going back and forth, you said your groups use the ultracentrifuge. Yes. Love it.
That's the point. Depending, there are some people that prefer another methods. In our case, we have lots of groups using ultracentrifugation. So that's, again, there could be some pros, there could be some cons, and back again to what we said at the beginning and in the previous session. Whatever you've done, you're doing it constantly into your samples. So if you are not able to exclude one particular subpopulation, that subpopulation will be still there forever for all your samples. It's only the one that you're interested that is appearing or disappearing, the one that will make the difference. Now, in terms of how much you want to remove that population, that's a different story. If you want to get a super pure population and remove all the lipoproteins, et cetera, then you need to change methods or a combination of them, which is another option as well.
Okay, so you need to make sure that even with size exclusion chromatography, you get the different sections. So you do all the different elutions there. You end up with, 13 of them or something like that. And same thing when you're doing ultracentrifugation. Each step gives you a different cell-free, then you get rid of apoptotic bodies and things like that, debris. But keep all those supernatants because we don't know what may be in them, so you can always go back. Once they're in the trash.
Waste too, the first waste as well.
Once they're in the trash, it's a little more difficult to go back and be like, oh man, look what I lost. Plus it makes an interesting story that you can also do it as a quality check, right? So how efficient are your speeds? Because now the protocols are pretty well defined, but it can vary between sample type and things like that. You almost really have to hone in and set your speeds correctly because not all centrifuges are created equal. It has its ups and downs. Same thing, the size exclusion chromatography, it's dirt cheap, but you have to keep buying the consumables. And then with that, you also have some, quality things that you worry about. I've noticed in our hands that sometimes 1, 2, 3, 4, great. You get all your exosomes in there and then your proteins later. Other times it moves down to be like 3, 4, 5. It's just dependent. So you save everything, you check everything. It's all about quality assurance.
John, one question. You have mentioned, and as always, you're correct. You have mentioned that you have different to keep all the supernatants. Would you recommend to one person that's starting any kind of project to make different speeds and get the supernatant at different time points at different speeds to check? Because one thing is what is published, and the other thing is what you're facing. And not all the instruments, even they are sold as unique and they're all equal. Just by working with cells, you can see when you centrifuge at this speed for this amount of time, they don't look the same. So would you recommend to have like a pre-analysis first?
Yeah, there's got to be, you're doing that. You want to, like I said, you want to kind of figure it out. So yeah, I would definitely sample off. I guess that's where some people are trying to move away because of the time consumption portion. ask anybody, and they want to do things as quickly as possible. And then as efficiently as possible. Time is money, and you see it that way. I mean, if you can do something that used to take you a day, you can do it in an hour. I mean, you look at that, and over time, it's like, hey, now I can publish quicker. So that's why, like you said, some people are diehard on it. Others are just, hey, what gives the same type of result but faster? I mean, I've seen all kinds of methods, people trying precipitation type methods and density gradients and things like that. And it's just about trying to separate out. It's mostly the exosomes from the proteins, right?
Mm-hmm.
And so that's the biggest step. I mean, getting rid of the cells, I mean, that's not that bad. That's in the regular portion. I mean, people do that all the time, but they used to get rid of all this stuff, which was thought of as junk. And so you go into that aspect. So same thing is, no matter what you're using, you keep the sections, you test the sections, and then eventually you come up with what works and what doesn't work. There's usually a list of doesn't work and a work. So
Correct.
Yeah. So it's trial and error, but hopefully, like I said, there's a lot more published, so at least you can get into an area, set you in the right place.
But there's a lot of things published and they're not right. So I guess the thing will be to go closer to what we are now, 2019 literature in 2016, 17, 18. Right?
Yeah, I've heard of actually working groups that get together to discuss best isolation method or best way to look at, the microRNA when they get to it. And one of the funny stories is someone was telling me is that there was a group of 5 and they were tasked with coming up with a consensus isolation method for the group, the entire group. And when they came back, they each had 5 different methods. So their one job was to come up with a method, and 5 people came up with 5 methods. So, so it's a little, it's funny in the sense that, that's the way it is. And that's why people say, no matter what you do, keep it the same and report it. So if you're using an ultracentrifuge and you have your basic side angle, or, however your actual rotors are going, mark it down, say what it is, test the levels of the sample. So the supernatants, say what's in what. And so that becomes, I mean, each step of this has so many weird places to trip up. But like we said, you're not necessarily tripping and falling down a hill. You're just tripping, you're getting back up, and you're moving on. And the sad part is you have to trip every time. So if you trip once, you got to trip again.
Because again, the other thing that is affecting it as well, the sample preparation, is the tubes because of the shredding. So you have different kind of tubes. Do you have some favorite ones, some others that are less favorite ones?
Well, from what we've learned, we've tested a bunch. And if you look at the paper by Danielson and Gaenna, in +1, it's about diurnal variations, but we put a bunch of steps in there of things that could go wrong, such as shedding of the plastic. So we tested a bunch of different Eppendorfs and found that a particular type worked better. They didn't shed, so because shedding of the plastic will add a very beautiful, approximately 80 to 90 nanometer population.
So that 80 to nanometer population went away when you changed the different tube types out until you got to the set? Correct.
So we figured it out by accident. It was just, so what we did was to figure it out was started centrifuging the PBS with everybody else. So, general scientific, great scientific method, right? Just run everything exactly the same. So we just kept taking the PBS, putting it into a bunch of different tubes, spinning them all, and found one that we didn't get a weird population in, and said, well, N of 1, perfect. Let's just start there. No, we did it a few times, made sure. or we went to Kinko's and just photocopied the results. So the great scientific method. But no, we found one. It worked perfectly. And then we found two with the flow. You're okay with different solutions. You're not really doing anything hard, but we did go through, tested two to make sure. And, obviously glass tubes are the best. No shedding there. They're perfect. But other tubes were fine. Yeah. I mean,
And do you work
sometimes
with 5 mLs or 1.5?
1.5.
1.5, yeah. Because
Yeah.
we're not working with so much sample. That's a good thing of flow. We're not
Yeah.
using too much.
Yeah, we're gonna.
Of the precious sample.
Yeah.
I think we get into that a little bit too, right? That's what makes flow glorious. Not just because we're a bunch of flow people who are going to preach that flow is awesome and that you should come to our labs and spend lots of money. I mean, time and effort.
Time.
So, but obviously we have to acknowledge, the front side, because I believe it was a gentleman. Oh God, what was his name? Alfonso. Am I saying this right? Blanco, who in the steps of flow cytometry put fluidics at the prep point. Not just the fluidics within the machine, but the fluidics becoming part of prepping the samples. Because if you don't have good preparation, nothing else is going to matter. You can put it in whatever machine you want. It's not going to fix the problem. It's just going to show you the problem. So, and what's our other phrase? The machine doesn't hate you.
Yep.
Yeah,
Really?
that is.
Said that before, probably going to say it a bunch of times. So, other ways that you could do it, Jenna, I've seen people do kind of like affinity-based assays, where you're doing some sort of capture to it, almost like a bead column, right?
Okay.
So you can do that. Listen, you start reading the literature, the microfluidics has become huge, right? Chip assays, to try to do this stuff, but still the gold standards, ultracentrifugation, size exclusion chromatography, starting to creep up the list. But even second place, there's this gap, right? And a lot of that is probably just because people went out and spent the money on an ultracentrifuge too, started the projects, and now aren't going to change because, like you said, you'd have to do the old clinical thing where you go, okay, I'm going to use both methods and prove that there's not huge differences.
Mm-hmm.
And we know there are, so that's not going to work.
Okay.
So again, Alfonso was very nice to point out, and I'm sure he's going to say it a million times, is you got to watch out for contaminants here. So don't keep using the same PBS, even if it's in a hood, it's going to get funky. Yeah, contamination. Obviously don't lick your fingers and touch the tube.
It's one of the key things. You will see a lot of things happening, and I remember one of the users that was using the samples, and suddenly their cells, I'm talking now about cells, they were starting to die. And we didn't know why. And suddenly I thought on the buffer, is it fresh? Well, yes, it's fresh 2 weeks. Fine. Look at the microscope. And you can see a lot of things growing on their PBS, on their fresh PBS 2 weeks old. So have a look to any source of contamination, because we are looking at very, very small things now.
Okay. And I thought that you guys also recommended that when you're doing this to keep a sample of the buffer to run as a base of it as well. So that way you would see it if there is going to be a contamination issue in that day.
Yeah, that's the thing what we were saying exactly, is run the sample with the buffers. So anything you do for the sample, do with the buffers. So centrifuging, or like you said, you get the different, when you do the size exclusion chromatography, which we do, is we get that first flush and stuff, you keep that, you run those, you run the buffers with it. So any of the buffers that you use, you can run. So even with your sheath fluid. And then I know Alfonso talks a lot about bubbles. So
yeah, the different bubble impacts of these micro bubbles on the sample. How do you count for them? Or do you just do the same thing? If you vortex, you vortex?
Yeah, but for how long do you vortex? So depending on what you have. If it's,
Yeah.
I will be vortexing. That's it. If it's Wednesday and I have a discussion with my boss, so you have a
Yeah.
good variation between
Mhm.
the vortexing. So I prefer not to vortex the samples.
Nope.
In fact, some of the instruments might steer, so I turn off the steering option. Pipetting, gentle, be gentle, up and down. Love your sample, kiss them. Okay, no kisses nowadays. Okay, up and down. All right. And the other one is just spin to one side and the other. But what is important is that you minimize the amount of air bubbles in your tubes, but also in your buffer. I prepare my buffers because I want to have extremely clean sample. But what we do is we prepare them at least 1 day before, with bacteriostatic. Okay, so we want to make sure that we don't have any air bubbles. The same thing that what we do with some the PBS or the sheath for sorting. We just leave them overnight. Bubbles are gone. Okay.
Okay.
You have another way, John?
No, I mean, that's probably the most efficient and easiest way.
Cheap.
Yeah, cheap. I mean, let it
24
sit
hours set.
let it sit overnight. Yeah, it's not a lot of effort going on there, which we like. So, and you don't have to sit and stare at it. It's not like paint drying, right? So that's good. So we can take care of all that stuff. All right, so Jenna, let's other things that your people have to think about. So let's say they got to this point, everything is good. They've taken all your advice.
Oh, okay.
Yeah.
This is a really good, let's say.
Forget it.
This is science in a vacuum, right? This is we're all isolated, so we can dream. That's all we have left, right? It's just a dream. So they've been perfect. So now we have to figure out, okay, how do we size them? What do we do? So there's many different methods to do that.
Okay.
Or you could ask, hey, what are those methods? Glad you asked, Jen.
Do you recommend some form of like bead controls, or do you have to have certain biologics?
So, biologic stuff, we'll get to that stuff because we're going to get
Okay.
to the flow. Yeah, we got to walk slowly.
Slow
Because you
down.
need,
Slow down.
yeah.
We'll right to that point.
Hold on. I get to do this. This is awesome. Buzzword alert. You need orthogonal methods. So in the world of EVs, the word orthogonal is thrown around so much because you need all these. And for the layman like me, I had to look it up. It just means different, but it's fancy. So, so basically, we have flow, but there's also people do Western blots, PCRs, things like that. But you're talking with those, you get just kind of a bulk analysis, right? That's simply, this is all going into our plug of cytometry. So what's the other one? Spoiler alert. We're going to flow. These end up, they just give you an idea, right? It's just like, hey, here's the Western blot, and that showed that we have exosomes because they're positive for X marker, right? Same thing with the PCR. Oh, look, we have stuff there. It's proving our point. But it tells us nothing about the individual exosomes, EVs. It doesn't tell us anything about cargo or content. And then it depends what your lab has around. A lot of people did have just sitting around like DLS machines, dynamic light scattering. But the problem kind of a little bit with that is light scattering. It's kind of what we do. Yeah. So how orthogonal are we talking? If you sat there and you said, well, the microscope, the flow cytometer, and the DLS gave me the exact same answer, someone may raise a hand. Yeah. Alfonso's already doing it and say, well, you're kind of using the same method, to see it. So it's good that it's consistent because at least, using the same thing, you didn't get different results. But, so they've come up with all these other devices, NTA, nano tracking analysis. Then there's tunable resistive pulse sensing. And then another company gave it a different little twist on it, which was more of a membrane. You can measure zeta potentials. You can do all these types of different little analysis. And then again, gold standard still is microscopy, you know, electron microscopy, the reemergence of dark field. Sometimes in my basement here, I feel like I'm in that, especially with that. So
Get in there.
yes, I am the exosome in the dark field. So I guess that's show and tell.
So all these orthogonal methods that you have to do on top of flow cytometry as well to prove the point.
Jenna, think about this. We are talking microvesicles, and the question is why your user wants to do it. They want to identify something. They want to identify what is causing a disease, or how effective is one drug for a treatment in particular. So you have two options, or let's say more options, but one of them is to know if you have something different. The other thing is to know what you have different. So usually I said, if I look at different fruits, I can look at them and count them, or I can make a smoothie. And this is what they're doing usually in Western blots or proteomics or genomics. They put all the fruits together, they mix them up, and suddenly you taste it. Oh yeah, that's good. It's a good flavor. But there's something there, and you don't know still what is that extra flavor that is giving you that difference, and where it's coming from, and what is causing that. If the presence of that extra fruit is too small, sometimes you will not notice, but it's still there. It's only when it's slightly more intense, when you know that it's there. So you have to go for a more sophisticated analysis where you will see and analyze each one of them independently. If you think on cancer and tumor samples now, they're going for single cell genomics. And that's because it's quite heterogeneous, and we want to know exactly what is the difference, because that will make the difference on a treatment A or a treatment B. So I guess the point here is, with flow cytometry, we can analyze every single one of those EVs. Well, probably not single, they could be attached, but ideally and perfectly would be one single event. And it's telling me that we have one subpopulation exactly different than the others, okay? So that's the beauty of flow cytometry, plus the speed that we have.
So
With
the
the
others
speed that
aren't
we
the
have-
best, but flow cytometry helps to identify more of the separate ones.
Yeah, and the other thing is that we can start to go faster and analyze and make a high throughput. Later on, when I see the difference, I need to prove those differences. And then I will go to electron microscopy or confocal or another methods to prove I'm working with EVs. These are perfectly, and these are the different samples. And this is what is happening in this one, and this is happening on this one. But I can prove it with the other technologies. But I will make the first screening first with flow cytometry. That's what I usually recommend.
A lot of people do it the other way. It's not bad. You can do it in both sections. I think originally a lot of these things were thought, well, if I didn't know the size, you couldn't really go for it. And even with flow, that's still kind of a thought, because it's like, if it's below the level of detection of what your instrument can do, then why even bother? But sometimes it's not so much about, hey, can I detect all of it or not? We're talking about very heterogeneous populations. I think the correct term is limit of detection. What is the smallest you can actually see? Because you're going to see something, but it's obvious you're going to lose, and it's just, where is that point of starting to lose things? And then the same type thing is DLS is great, NTA is great, TRIPS is great. They all add to the story, Western blots, PCRs, because they each have their own benefit, right? The problem being is that you can only do lower numbers, right? It's not high throughput. And a lot of these will tell you zero about the cargo. There are some capture assays that have started doing that a little different, where you capture the exosomes on a substrate, and then you can come back and label with something else. So that can give you a little bit of an idea. So if you have something that's like a protein marker or something like that, you start looking like Alex or something like that, calcium type things. It depends what you can label with. And they are working on being able to actually get things internalized, even though they're stuck on the substrate. You still label, you let it sit, and things go into the membrane. So, people are working hard to come up with solutions. And so that's starting to be a little bit better, but the throughput is still low, right? It's just, you put a sample, you wait because it's got to bind, then you wash it all off, then you add something else, and you get a reading.
Mm.
But you only get so much.
The other thing is, as well, Gaenna, in flow, you have two options as well. You have the possibility of doing flow cytometry to identify different subpopulations, or you can use as well beads to capture all the exosomes that they have one particular marker. Once you
Okay.
capture them, you can do the analysis of quantification. How many exosomes I have here? Okay, I can do it with Western blots, but by flow cytometry, you could be far more accurate, and it's more sensitive than the Western blot, and you can quantify it by doing calibrations.
And then does sample size have to come into play at any of this between all these different multi-pair matrix? Do you have to have a certain amount for each, or is there a benefit to just doing flow then going into something else?
The amount of sample that we spend is nothing compared to something else.
Yeah, compared to it all, and then you end up doing dilutions, right? So, like Alfonso said, if you start with the flow cytometry, oh, we finally got there. And why wouldn't you start with flow? Let's be honest, right? So when you do that, there's the swarming, right? So Edwin van der Pol put out the iconical paper on swarming, or it's a form of clustering, right? So having multiples come through a laser at the same time. You got to remember that compared to these little guys, the laser looks like a Mack truck, right? It's huge. And basically what they're doing is saying, okay, you have to do serial dilution in order to make sure that you stay linear. Once you get off that plane, it's probably just a bunch of things being put in at the same time, or they could be fused. So if you do the dilutions and it never stops, then somewhere in the process, you ended up fusing the exosomes. So you have to be able to see a little bit, and it's simply the dilutions will tell you that. Multiple papers after that have come out doing the dilutions. So by the time you're done, I mean, we're usually at a 1 to 1,000 with the red blood cells, and that gives you sample for days, right? I mean, we did a 1 to 1,000 on a viral particle, and I've been running it through the flow as a control for like several months. It's still sitting in my fridge. There's still plenty, because like Alfonso says, I mean, how much do you need to run to get an idea if it works or not, especially on a quality control? And so you have that. And I think the flow part also, there's so many other possibilities. So, multi-labeling. Let's not get confused though. We'll get to that part. You can't sit there and go, well, I'm going to put 10 colors on my exosome.
Please don't do that. Please.
Yeah. I mean, it's like sitting there saying that, the three of us are going to be happy sharing one M&M. It's just, it doesn't make sense.
I'll take it.
Yes.
Okay.
That's a good point. You just made a funny point. People may not be able to see, but out of the three of us, Alfonso is the biggest. And so if he gets to the M&M first, none of us are getting M&M.
No, forget it.
So if you're a huge molecule and you come on an exosome, no one else is going to play.
I will not be polite.
No.
No.
They just block you. You have no choice. If he wants to keep his M&M, he just keeps moving around, and you're like, come on, man.
You release it.
No.
Yeah. Oh yeah. So M&M, feel free to send some money for endorsement. There we go. Not even realizing I'm basically, I'm plugging the M&M company.
Jeez, brand.
Yeah.
Brand.
Anything Skittles, a peanut. Yeah. So that's a very good point too, is that, once you start, you got to go with it. And then there isn't a one size fits all on this, right? So no one's come up with markers that work perfectly. We already talked about how the tetraspanins are great for most, not perfect solution. You have to use them as a cocktail. And then red blood cells are one of the particular types that do not have tetraspanins. So you have that problem going. And so I think on the next session, we're going to talk a little bit more about the flow cytometry aspects and setting it up. And so we'll
Okay.
really touch base on what you want to do. But to kind of finish this up is one of the big things. If you listen to a lot of the talks, they talk about kits and stuff that all do membrane specific stuff. So if you can label the membrane, it's going to give you a lot better idea. The fluorescent proteins start to work. People start labeling with the fluorescent proteins. There's been talks of CFSE, but you can see there's pluses, minuses. If you don't have enough enzymatic properties there, you'll never flip the switch. So you get kind of not a whole picture of what's going on. So people are trying to find something that doesn't take a lot of other things to happen.
Okay.
To show positivity. So like we said, the lipophilic membranes, all right, it's a lipid. And lipid dyes tend to show some of the story. There's cell trackers and I mean, every company has some sort of membrane dye, right? You can find and go with it, and in all different flavors too. So if you have something that's already GFP labeled per se, or has a FITSI tetraspanin cocktail, there's reds, deep reds,
Anything we could do to focus on maybe some of the intercellular cargo on top of being the membranes?
Yeah, there's some protein dyes that'll go in. Dr. Guérin's group, Anita's group, he's along with the NIH and stuff, they're playing a lot with these labeling of the microRNA. They put out a paper based on molecular beacons. And so if people want more detail on that, I think that's above the scope of what this little small talk is.
Right.
But it's awesome because you're able to, not just like a Cyto 9, you're not just labeling everything inside, the RNAs, the DNAs, everything that happens to be there. You're specific to a particular microRNA. So you go through, you find your libraries, you understand what microRNA is associated with that type of cell, and then you design the beacon towards it. There are loop sequences that are matching pairs. And then as soon as they bind to that microRNA, poof, they light up like Christmas trees.
The other thing, because you're mentioning now, John, all these markers, just quickly, there's a lot of people using thresholding on scatter. And because my particles will have this marker, I will do the thresholding using this particular marker. I think it's a risk. Use a threshold on that particular marker, because you could be losing some other stuff that is quite interesting, even more interesting than those particular microvesicles that you're looking for, or exosomes that you're looking. Do you work more with violet side scatter, blue side scatter, or do you prefer to work with triggering based on the signal that you're looking for? Because you could be completely wrong, and not all your EVs are showing or expressing that one. Or if you're sorting, but you're not looking as well as some other particles that could be there.
I think what we're going to do is, we'll get into this deeper. I think in the next section, the way we've broken it up and how we want to do this. But to touch on it, what we started doing in our experience is now, I'm a scatter guy. For the longest time, no one wanted to label because there was not a good label. It was all tetraspanins, and we have red blood cells, again, no tetraspanins. So we started just playing with scatter properties. We started a lot with using, our sorter and starting different protocols there, which laser lines worked, which didn't for scatter properties, because we're able to use all the lasers lines that we have on scatter. On top of that, once we identified some populations, I also started using a fluorescence trigger. And when we started moving that way, and using the membrane dyes, and basically I record both ways. So I'll start with scatter so I can figure out what I'm actually looking at. So I'm not just, putting on the blinders, and I know a lot of people do that. They look at, figure out, and then work backwards. So I get two different looks at it. And then, a lot of times, while percentages change, profiles do not. And it's, a lot of these things is, as I told Jenna before we're doing this, is you become very paranoid. So I know a lot of times when you get positive results, you're like, yes, it worked. In this, I've gotten to the point where it's like, hey, it worked. And the customer's all happy, the researcher, and you go, yeah, I don't know. And they're like, what do you mean you don't know? I'm like, hold on a second. Right? Let me try this. Let me try that. I'm going to use this scatter. Then I'm going to use this, fluorescence trigger. Oh, look over here. What is that? And you get to the point after doing this for like 8 years that I don't trust myself anymore. Like I am so honed in on proving that it's right. And a lot of times when I used to work with a gentleman, Bacillus toxovides, I'd sit there and we'd almost do it separately to say, okay, I just did it. I'm not going to tell you what I got. You go do it. And then we come back and go, good, we got the same thing. Or, okay, which one of us messed this up? Or is it
definitely
yeah, or is it so off that it's inevitable that you end up with different results? So, you put all these things in because you just keep scratching your head. You're just like, okay, I don't want to give out misinformation and look like a fool. Okay, more than usual. So Jenna, if you're not a compulsive person at the moment or paranoid, I know you're a little compulsive, but paranoia will set in, and you'll just start doubting yourself because it's gotten better. It's gotten better. I've relaxed a little bit. But in the early stages of this, trying to develop protocols, everything you published, you got a little nerve-wracked, right? Because you're so afraid that the powers that be and the people that are working on this every day know something you don't already, and that as you're in the field and you're just doing it and things are coming day by day, updates, you're just afraid everything you did is just going to be like, oh nope, that's all wrong.
That's outdated.
Yeah. But
so
to get back to the
key
whole
take point was to keep it consistent, it sounds like.
Correct.
Yeah, always.
Yeah.
The same. And then, like Alfonso said, is consistency 1, and 2 is don't be afraid of multiple methods in order to get a decent picture. So all you're trying to do is make sure that it's not about just, okay, going and running to first base is sufficient. Cover all the bases, each and every time. Maybe you get more information as you go along, but at least you've done due diligence. You've looked at everything you possibly can, and you're giving results to the best possible, scenario.
You have to be as critical as you can, always. And you have to be, as John was pointing out, you have to be as critical as you can with yourself, but as well as we mentioned already with the papers that they are published. Okay, not everyone is using everything correctly, well, except John. But usually you will see mistakes, and even in the same group, you give the same protocol, you will see mistakes, and it's because of the way the people is doing things. I mentioned already pipetting, for instance. If you give the pipette, there's more energetic people pipetting up and down, and less energetic people. So you can see just pipetting will create a mistake. Timing, we will talk about all the different controls, but time is one of the most important ones as well, again, because you will have degradation, you will have time, as you mentioned, vortexing. For how long would you vortex? For how long would you keep your samples stored? So time is one of the key things here. As well, you will see time in the flow. We want to make sure that the instrument constantly remains as stable as possible. But you will see variations, not just in flow cytometry. You will see variations in every single method that you use. So they have to be consistent, not just in flow. They have to be consistent in proteomics, in transcriptomics, in electron microscopy, atomic force microscopy, because they will make mistakes as well in their sample preparation.
When you're setting up your baseline, then would you recommend then doing like different time points throughout the scale to see how things are changing?
Oh, yes.
Like
yeah. But in fact, we when we are working with groups, for instance, now we are talking about the COVID, for instance. Okay, we want to know when one patient arrived to the clinic and at the different time points, how is the evolution of that patient? If they are given this treatment, how is this affecting the person, but also how it's affecting my EVs during that period of time? Because they could tell me probably way in advance how the response will be. Okay, so I will definitely look at time as one of the key parameters, not just, again, not just for flow, for every single things that you're doing. We have been mentioning different essays. With every single one, I would try to make sure that all the users are consistent. Because the first thing they will do is Western blot. I know how to do a Western blot. No doubt I know how to do it. Yeah, but I know what I'm doing. Why are you sure the Western is the correct one and not the flow, right? Because you'll see we'll put a lot of controls, all right? In Western, how many controls do you have?
Yeah, I've gotten used to having an entire rack, and people are like, oh, look at all the samples. And you're like, yeah, look, I have a rack of controls and I have 2 samples. So it gets kind of funny at sometimes. And then
and again, electron microscopy, gold standard. What will be the picture that you select? The pretty one. You'll spend the full day taking one single picture to a couple of exosomes to prove that you're working with exosome. What about the rest of the sample? I couldn't see anyone, but I've seen 2 exosomes. Fantastic, statistically significant.
yes,
But it's the cover of the paper.
yeah, like I said, with the microscopy too, is that was the other thing we saw. So that's one of the ways we found out about tube shedding, right? So we did that. We did atomic force, and we could find things. You also can find filter pieces. So if you're using a filter more than once, or you're like Ianita had shown us on this particular instance, and Alfonso was saying, you come back and you're like, through the filter, I'm mad. My PI just told me I need to work Sunday. And then all of a sudden you see the pieces of the filter in there. They make nice pictures. They look like honeycombs. And you're talking about pieces of it. You can even get it from the instrument. If you're,
sheet line?
Yeah, through that actual, so, you have the normal maintenance of the machine, but it's almost suggested to change the filters more regularly. You can even just keep them and put them over somewhere else. So, but more of that talk's going to come about in the next section. We're going to do just flow cytometry, finally in the wheelhouse. Just going to close it up with a couple of things. Number one is, if you want to be the smartest person in the room when doing extracellular vesicles, stop now. Because it's best to collaborate with as many different people as possible, because there are all kinds of experts, and even those experts have particular things that they're experts about in the field. There's no overarching type of individuals. You have your physicists and your mathematicians and your scientists and your doctors, and everybody's got that piece that makes this puzzle a little more complete. There are great groups to join, out there, all kinds of new extracellular vesicle groups. I'm sure when we finally get back to some meetings, you're going to meet a lot of these people and hear their take on things. And so I'd like to thank everybody again for joining us. Hopefully our rhetoric is is not driving you crazy, and it gives you a little bit of joy, as it does for us. Of course, I get to sit here and just talk to my friends. So what could be better than that? Again, when you have questions, please put them in. We have individuals who are going to put them together for us. And then the live session should be a lot of fun. We'll get people to join and hopefully put their own spin on this, maybe add a couple of jokes or two on this whole setting. And then please join us for for section 3, where I'm sure it's going to get interesting, because now you've got 3 cytometrists talking about cytometry finally.
Moving into it.
Yeah, so that's good. Thank you, and stay safe.
Yep, and home.
And home.